On the night of January 22, 1988, at approximately 12:45 a.m., a 29-year-old Caucasian woman was entering her car as she was leaving a bar. The woman was drunk and leaving to return to her home. As she entered the car, a black male came through the door next to the driver's seat, opened it, and punched the woman in her face several times. After that, he took her to a nearby site where he raped her and stole her jewelry and belongings. The attacker then departed, leaving the woman in her car. There were semen stains on the victim's clothing left by the perpetrator, in which the police had collected using a rape kit. However, the rape kit was not used during the trial, where a man was convicted. DNA testing was not advanced enough to rely on and identify a perpetrator during that time, so DNA was not used or mentioned during a trial. The police took the victim in and gave her a collection of mugshots in which to choose from, and at that time the victim chose James Calvin Tillman, who had only had a minor run-in with the law before, but had no past records of violence or any serious crimes. The victim identified him as the perpetrator, being the only eyewitness. Tillman was 26, living in a homeless shelter and working at a car wash, when he was charged with abducting a woman as she got into her car near a Hartford restaurant, then beating her and raping her at a housing park. Tillman continuously maintained his innocence but was convicted after a jury trial of kidnapping in the first degree, sexual assault in the first degree, robbery, and assault in the third degree on September 9, 1989. The conviction was based almost entirely upon the eyewitness identification of the victim, a white woman who was unknown to Tillman, an African American man. He maintained his innocence and rejected a plea bargain that would have given him 8 years in prison. Tillman appealed the conviction in the Supreme Court of Connecticut in 1991. Also, Tillman argued that the jury selection was not right because court clerks had excused a number of potential jurors due to financial troubles. He claimed that he was most likely to face this hardship and punishment because of the fact that there were no African American males on his jury. His defense argued that the woman's testimony was unreliable because she was drunk and her eyes were badly scarred after the beating and almost closed during the whole raping, so she couldn't have identified the perpetrator. However, the jury found him to be guilty, based on the woman's testimony as the only circumstantial evidence. Tillman's verdict was affirmed in 1991 and again in 1999 when he claimed ineffective assistance of the counsel before the Appellate Court of Connecticut. In 2007, the Innocence Project organization was an organization aimed at helping wrongly convicted men and women and took it upon themselves to reopen cases. At that point, they viewed Tillman's case and decided to reopen it and get an appeal using DNA that was found on the victim's pantyhose. More advanced DNA testing were performed on the crotch sample of the victim's pantyhose and on multiple semen stains on the victim's dress. Although 99.9% of human DNA sequences are the same in every person, enough of the DNA is different to distinguish one individual from another. That is why DNA profiling uses repetitive sequences called variable number tandem repeats, particularly in that they use short tandem repeats. The DNA profile technique was first reported in 1984 by Sir Alec Jeffreys at the University of Leicester in England. So in this case, after the semen is collected, a reference sample is then analyzed to create an individual's DNA profile. They do this by extracting the DNA from the sperm. This is done with a mild detergent that bursts non-sperm cells, or a rinse with the water removes broken cells, and then a stronger detergent used to burst the sperm and recover DNA. The lab then compares the DNA from the semen with the suspect's DNA. A blood or saliva samples from the suspect will supply enough DNA for the comparison. Researchers are able to produce millions of copies of a specific DNA quickly with a method called polymerase chain reaction, or in short, PCR. The PCR starts with heating it to a high degree of approximately 96 degrees Celsius to separate the sample into a single strand of DNA. Two primers are then designed to match to the segment of DNA you want to copy. Through complementary base pairing, one primer attaches to the top of the strand at one end, of your segment of interest, and the other primer attaches to the bottom strand. When DNA is extracted from the semen, then it is identified and compared to the DNA of the perpetrator. And so when the semen's DNA was compared and identified to the DNA of Tillman, it did not match, exonerating Tillman from the crime. Another DNA profiling system is called the restriction fragment length polymorphism. The RFLPs are differences in lengths of DNA fragments after digestion of DNA samples with restriction endonucleases. These differences in lengths are caused by variations or polymorphisms in the DNA sequences in the human genome. RLFPs can be used to trace inheritance patterns, identify specific mutations, and for other molecular genetic techniques. The restriction enzymes added to the DNA being analyzed and incubated for several hours, allowing the restriction enzyme to cut at its recognition sites. That is why the DNA is then run through a gel which separates the DNA fragments according to the size. You can then visualize the size of the DNA fragments and then decide whether or not the DNA was cut by an enzyme. Although the RFLP is very well used, there are some limitations to it, such as a large sample is required to run an RFLP. So without a large sample, it cannot be done. The technique detects the lengths of a cut DNA. DNA that is heavily damaged cannot be used, and if only a small sample size is available, RFLP may still be usable if coupled with PCR, and PCR can amplify a small sample into a larger one. However, contamination of sample may cause amplification other than the desired DNA. Losing DNA samples of previous criminals and collecting it again would be a hassle. That is why the FBI, or the Federal Bureau of Investigation, uses a system that combines CODIS, or in other words, Combined Index DNA Systems. This is a computer system software program that operates local, state, and national databases of DNA profiles from convicted offenders, unsolved crime scene evidences, and missing persons. By doing this, the FBI is linking serial crimes to each other and identifying subjects by matching DNA profiles from crime scenes with profiles from convicted offenders. This is done because a criminal who commits a crime is most likely to commit another, making it easier to catch them. On June 6, 2006, Tillman's conviction was overlooked after the Superior Court granted a petition for a new trial based on DNA evidence. Tillman was released without bail. Further DNA testing was performed on one more stain on the dress, which was also originated from the source of other stains. On July 11, 2006, the charges against Tillman were dropped, and James Calvin Tillman was exonerated after spending more than 16 years in prison for a crime he did not commit. Years later, when the actual rapist was found, a judge remarked that the only thing Tillman was guilty of was an uncanny resemblance to the perpetrator, so the victim's mistake must be understandable. The justice system's mistake was not, however, which is why the Connecticut legislature passed special legislation to provide compensation to Tillman after his release. James Tillman has been serving as vice president for the Ministry of Hopewell Baptist Church since his release. The government and the governor of Connecticut offered him $500,000 in compensation for his wrong incarceration. Several members of the state legislation have introduced a bill to provide Tillman with a total compensation of $5 million for the 16 and a half years that he wrongfully spent in jail. On May 16, 2007, the Connecticut legislature unanimously voted to present Tillman with $5 million to assist him in living comfortably without his wrongful imprisonment. The legislation stated that they were touched by his attitude following his release.